What if we were the last generation of humans to evolve naturally? Not because evolution stopped, but because we decided to take control of it ourselves. Innovation is improving faster every day, and it's only a short amount of time before our handiwork catches up to us. My name is Irmak, and I'm a grade 11 student at TEDxWAB, and I've been interested in genetics since middle school. So much, in fact, that I want to study it in the future. But the more I've learned, the more one question keeps coming back to me. Just because we can change DNA, does that necessarily mean we should? That's a question that lingers in the back of my mind whenever I think of genetics and genetic modification. Right now, we're living at a turning point in human history. For most of humanity, genetics and our decided genes were something that happened to us. They were predetermined for all of us. We inherited traits, diseases, and variations without any conscious external human control or manipulation. But now, for the first time, we're starting to decide what stays and what changes in our genetic makeup. Now, however powerful that shift may be, it's also being proven to be incredibly dangerous day by day. Genetic modification offers many improvements to agriculture and medicine. However, in the wrong hands, the line between personalization will inevitably blur. The reason for this is a technology called CRISPR. Scientists often describe it as a pair of molecular scissors. It allows them to cut DNA at a precise point, enough that people can compare it to editing a sentence, fixing a mistake, and inserting a correct word in said sentence. Now, the potential here is incredible. Diseases like sickle cell anemia, cystic fibrosis, or beta thalassemia are written into a person's DNA from before birth. Their treatments involve treating the symptoms of the diseases instead of the illness itself, which only manages these conditions and doesn't cure them. So instead of treating symptoms for life, we could remove the cause entirely. That's the power CRISPR grants scientists and doctors today. And it's already happening all over the world. In agriculture, scientists are creating crops that resist disease and require fewer resources to grow. Unlike antibiotics, which have been creating new resistive genes in livestock resistant to treatment, genetic modification such as CRISPR can allow for that issue to be entirely forgotten about. Additionally, mosquitoes are being genetically modified so that they can no longer spread malaria, a disease that kills hundreds of thousands of people every year, many of them being children. So if gene editing can reduce suffering at such a massive scale, that's amazing for the future of humanity. Only not quite, because that's not the problem which should concern you about genetic modification. The problem is what comes after the science. Because once we have the power to change human genetics, we also have to decide how far we're willing to go, and who ultimately decides to make that decision. In 2018, that question became real. A scientist edited the DNA of 2 human embryos to make them resistant to HIV. These were not embryos with a life-threatening condition. The embryos were completely healthy, and it was simply an experiment for the use of CRISPR. The experiment was done secretly, without any global agreement, and without understanding the long-term risks that was done to patients who did not need it. 2 baby girls were born, and the global reaction was immediate. Because how could these children possibly consent to a procedure which altered their DNA from before birth? A choice which may have changed their medical future forever, decided by their parents and doctors, and was ultimately unnecessary for their overall well-being. If it weren't for the experiment being conducted without clear limits, there wouldn't be the outrage that the public ended up portraying. The problem was never whether the science existed, only how that science was ethically applied. That moment forced the scientific community to face something uncomfortable sooner than we expected. The question of whether scientists can actually modify genetics was no longer in question. We're asking when we will do it, how we will do it, and who ultimately decides to go through with that procedure. And all of that is true. Who does get to decide? The patient, in which case human embryos cannot consent. The parents of the unborn children, predetermining their entire life, their doctors, their governments. who? If we can edit disease, it feels right to use this technology for the greater good. However, once that door opens, it's impossible to close because it becomes much easier to justify editing other aspects of our own humanity: appearance, intelligence, physical ability, and many more. When we reach that point, gene editing is no longer just about health and improving healthcare. It becomes about personalization and preference. And we've seen the school of thought before, and it was never for the benefit of humanity. We didn't see it as editing individual genes, but rather how and who expressed specific genes. Eugenics, selective breeding violently applied to our own species. In the early 20th century, the United States carried out forced sterilization programs where women were made infertile without consent because they were deemed unfit. This included women with disabilities, mental illnesses, people of color, or simply those living in poverty. These policies were supported by the law and justified as a way to improve society. It didn't improve society at all. It was decided by laws and bills written by men that sterilization did not affect. It was never decided by those forced into it. And on the other side of the world, almost 20 years later, in the Second World War, in Nazi Germany, this idea escalated into something far worse. Under the belief of a superior Aryan race, the state attempted to control the reproduction of people to create what they believed was their own superior population. The horrors of what the Nazis did, where they sterilized, persecuted, and killed innocent people, should never be repeated, and prove why such ideologies should never exist in the first place. And in other parts of the world, governments have always interfered with reproduction to control populations and weaken communities, and unfortunately, these policies have never been random. They come from the same belief system that some lives are more valuable than others, based on genetic markers or cultural markers like physical attributes found in different races, religious communities, and cultures. CRISPR isn't a means to erase that history. However, in the wrong hands, CRISPR gives us the ability to repeat history with far more precision at a greater, more devastating scale. Instead of stopping children from being born, science today grants us the power to alter them before they are even born. Additionally, what makes this even more complicated is that we've always influenced our genetics in one way or another through epigenetics. Epigenetics, essentially, is how our environment can change and changes the genes that are expressed. Stress, nutrition, and life experiences are all factors that can affect which genes are active over time. Identical twins are often used as a prime study to show how drastically different epigenetics affects their lives, as they start with the same DNA, but their lives can lead them down very different roads. For example, imagine two identical twins. Hypothetically, one twin had a more stressful life, chain-smoked, drunk heavily, had more sun damage, and took on more laborious jobs and had more exposure to experiences which negatively affected their health. Whereas the other twin did the complete opposite. They aimed to live a mostly stress-free life, minimally smoked or drank, took on desk jobs, and therefore were not exposed to these events which did not negatively affect their health. Although twins are epigenetically indistinguishable during the early years of their life, as time goes on, epigenetics plays a key role in developing genes and traits which change the outcomes of their lives. But there is a key difference here between epigenetics and genetic modification, such as CRISPR. Epigenetics is not controlled by a single decision. It happens naturally through interaction with the world. CRISPR is different because it is intentional. Someone is making a choice about what should be changed in the person's genetics and how. And once that choice exists, a secondary issue appears as well: access. Gene therapy is already very expensive and limited. However, in the future, if it becomes an acceptable practice to genetically modify children, it could increase inequality in a way we've never seen before. It would not just affect opportunities, it would affect biology. Some people would be born with advantages others could never dream of accessing. And this brings us back to the central idea. Without clear limits, gene editing does not have the potential to improve lives. It has the potential to divide them and repeat the same thinking that led to eugenics. And therein lies the difficult questions that humanity must answer. If parents can prevent fatal diseases, should they? Most people would say yes. But if parents can choose traits like intelligence or appearance, should they still have that right? A child cannot consent to being genetically edited. So do parents have the right to decide their own future before the children are even born? And if we start removing what we see as flaws, what happens to diversity? From a biological perspective, variation is what allows populations to survive. It creates resilience. A perfectly optimized population might seem ideal, but it can also be much more vulnerable to change in an ever-changing world like ours today. When we remove limits completely, we do not always create something better. Sometimes we create something even more fragile. So will we be the last generation to evolve naturally? Probably not anytime soon. Gene editing, like CRISPR, is strictly regulated, even in agriculture, and not widely accessible to human patients. But for the first time, it is possible for humans to guide their own evolution. And that brings us back to the question that matters most. Not whether we can change our genetics, but who gets to decide what those changes should be. Because if we do not answer that carefully, we are not just shaping the future, we're risking repeating the past with the power to make those decisions even more everlasting and permanent. Thank you.